Good morning, everyone. It's nice to see you could make it out this early session here. I'd like to thank Adriana for inviting me to be a part of the symposium. So, outlining my talk today, first I'll go over the theoretical importance of brain signal variability in general before describing a few different ways we can measure bold signal variability in fMRI studies. Then I'll talk about some task-based and resting state research that have used bold signal variability to characterize typical development and cognition. And then finally, I'll present data from a couple of studies that have used bold signal variability to characterize atypical development in ADHD and ASD. So first off, variability is not always bad. It can be beneficial and not detrimental. So on the neuronal level, we know that neurons do not fire in the same way twice to a particular stimulus. Now, one theory why this might be beneficial is that this could support adaptive behavior, because if your neurons fired in the same exact way to every stimulus every time, you can never change your behavior to that stimulus in different contexts. Additionally, simulation work has shown that network responses formed with noise are less susceptible to disruption, so they're more reliable. So on the micro level, it seems that variability may be beneficial for adaptability and reliability. On the macro level, which we study using fMRI, it's thought that increased variability can facilitate network configuration flexibility. So you can see here a hypothetical 3-node network. This is some simulations by Randy McIntosh. And you can see here on the left, if these 3 nodes have very low amounts of variability, the amount of network configurations they can enter into is very sparse. On the other hand, you can see on the right-hand side, that if these 3 nodes have a large amount of variability, then the amount of network configurations they can enter into exponentially increases. So on the micro and macro levels, it seems that variability could facilitate adaptability, reliability, and network flexibility. So how do we measure bold signal variability? Well, the most obvious way is to simply calculate the standard deviation. So simply assessing on average how far time points are away from the mean. And this is a time invariant method, meaning that we could shuffle up the data points and the SD is not going to change. Another way to calculate variability would be to calculate mean square successive difference. And essentially, you're calculating the difference between each time point here and then averaging those differences across the entire scan there. So this is a time variant method, meaning that if you shuffle up the time points, MSSD is going to change. And then finally, we have sample entropy, which is actually a measure of predictability, but for purposes of this talk, I'll just refer to it as variability for simplicity. And it's important to note, though, that these two are not synonymous. You can have high variability, but low predictability, for example, a sine wave. But for the purposes of this talk, I'll just refer to it as variability. And in sample entropy, what we're doing is we're identifying repeating patterns within the data. So there are 2 parameters here. M is the number of time points we're interested in. In this case, or typically in fMRI studies, it's 2. So what you're going to do is take each pair of time points and compare them to every other pair to see if they match within a tolerance R. And that goes on your denominator. And then you're going to take M plus 1, which would be 3. So you're going to compare triplets of time points across the entire scan and see if they match within some tolerance R. That goes on your numerator, inverse log, and essentially higher values means less predictability or more variability for the purpose of this talk. So essentially, brain variability can be beneficial and not necessarily detrimental. And then we have different ways of quantifying bold signal variability using both time variant and time invariant methods. So now moving on to some task-based and resting state research. A lot of this task-based research has been done by Doug Garrett, and essentially he looks at older and younger adults during a cognitive task. And what he's shown is that increased variability is associated with younger adults who have faster reaction times and more consistent reaction times. On the other hand, decreased variability is associated with older adults and individuals with slower and less consistent reaction times. So this is a resting state study that our lab carried out. We looked at bold signal variability, how it changes across the lifespan. And we used the NKI data. We had 2 groups here, a fast TR group and a slow TR group. And we calculated voxel-wise MSSD. And we used standard GLM model with age as a dimensional predictor. And right here on the right, the blue is the fast TR group, The red is the slow TR group, and the violet is the overlap between the two. And generally, you can see that most areas in the brain decrease in variability across the lifespan. There were a couple areas that did increase here in the dorsolateral insula and the ventral temporal cortex. But in general, what we see in task and rest is that variability decreases across development and the lifespan. So now we're interested in some test-retest reliability measures, seeing how reliable these different measures of variability are in fMRI studies. We used HCP data. We had 416 adults, and this data set is ideal because it provides you with 4 15-minute resting state scans. So they acquire 2 scans on the first day, 2 scans on the second day. And we calculated voxel-wise indices of each of these 3 variability measures. And then what we did was we calculated voxel-wise interclass correlations across all 4 scans. And then we also looked at what is essentially session reliability by looking at the 2 scans that were required back-to-back on each day. So first off, just looking at the spatial maps, the variability spatial maps, this is not the ICCs. And we see that our 2 time variant methods, MSSD and sample entropy, are essentially perfectly correlated here at 0.99. Excuse me. Interestingly, standard deviation is strongly correlated with both MSSD and sample entropy here at around 0.9. So all 3 of these measures are strongly related, and they seem to be picking up on similar things in the bold signal. So here are the test-retest reliability interclass correlations here. The first row is all 4 scans, and the bottom 2 rows are the 2 scans on each day, and you can see that the results are essentially identical, whether we look at 4 scans or 2 scans. And the important thing here to note first off is that all 3 measures, brighter intensities, are higher interclass correlations. And all 3 measures have very strong or large ICCs in the gray matter, as you can see here. They were all essentially around 0.7 to 0.8, somewhere in that range. Interestingly, what we see with MSSD and sample entropy is that they had very low ICCs in the white matter, whereas standard deviation had high ICCs in the white matter. We're not sure exactly what's going on, but it could possibly be that signals in the white matter and CFS are essentially oscillating, and perhaps the time invariant and time variant measures quantify those oscillations in different ways. But essentially, the main thing here is that all 3 measures are strongly related, and all 3 measures have high test-retest reliability in gray matter. So essentially, what we see across task and rest is that bold signal variability generally decreases with age. And then also all 3 measures, whether they're time variant or time invariant, are strongly related, correlated around 0.9 and higher. And all 3 have high test-retest reliability in gray matter. Okay, so finally I'll present data from a couple of studies that have used bold signal variability to characterize atypical development. So this is a study we did looking at bold signal variability in children with ADHD, with and without a diagnosis of ADHD. The average age here was about 10 years old. I think the range was 9 to 12 or something like that. And essentially, we calculated voxel-wise MSSD, and then we subjected that to a standard GLM. We did a categorical analysis, and we didn't find any group differences. But in a dimensional analysis, putting in ADHD symptom severity measures as a dimensional predictor, we see that regardless of diagnosis, increased variability is associated with higher symptom severity scores. So this seems to indicate that there may be an optimal level of variability. So good variability may facilitate cognitive performance, but if you go to the extreme side, then perhaps that is detrimental. So it's not always going to be the case that increased variability is always better. Maybe perhaps if you go to the extreme side, then that becomes detrimental. All right, and then finally, we have some data from a resting state study here that we did, looking at data from the ABIDE 1 and 2 data sets. This data was from the NYU site, and we had 57 ASD, 67 TD, 7 to 39 years old. We calculated voxel-wise MSSD, and then we used the standard GLM again, and we looked at the interaction term between group and age. And what we see here is that in several brain areas, individuals with autism, in the blue squares here, have a steeper decline in variability compared to typical individuals, which are in the circles here. So we saw that in this left superior temporal area, here in a right occipital parietal area related to the dorsal attention network, and then also in this ventral medial prefrontal cortex, part of the default mode. And interestingly, in the cuneus, precuneus part of the default mode, we saw the opposite pattern, where individuals with autism seem to increase in variability compared to this slight decline in typical individuals. So to wrap up, both variability can characterize atypical development. We saw it was related to behavioral indices of ADHD, but didn't differentiate between groups. And then also bold variability identified different developmental trajectories in ASD compared to TD, with most of those results showing that individuals with ASD had a steeper decline in variability compared to TD individuals. So to wrap things up here, what we saw on the theoretical level, brain signal variability can be beneficial, not necessarily detrimental. On the micro level, it's theorized it facilitates adaptability and reliability. And then on the macro level, perhaps it facilitates network configuration flexibility. And then I showed you research looking at task-based and resting state variability. And essentially what we see there is that variability generally decreases across development and into old age. And it's also positively correlated with cognitive performance in that task research I showed you from Doug Garrett. And then for the test-retest reliability, we saw that all three measures are strongly correlated. So whether they're time-invariant or time-variant methods of characterizing variability, they all seem to be picking up on something similar in the bold signal. And then all three measures also had high test-retest reliability in the gray matter, which is good news, of course. And then finally, I showed you data that characterized atypical development, and bold signal variability was related to dimensional measures of behavior in ADHD, but not categorical group differences. And then it was also differentiated between ASD and TD trajectories of variability across early development and into adulthood. So that's it. Thanks to my supervisor, Lucina Udine, and the rest of the BCC lab who helped out with this. And thank you for showing up so early, everyone.